Hi, I'm Sierra.
I'm Kate. I'm Anna. And this is Kristen. And, and we're looking at the cell cycle of breast cancer. So with breast cancer, we were looking specifically at the P53 mutation, which is a more aggressive form of breast cancer. So it often results in mutated, in like solid tumors. And the higher the levels of the mutation, the more aggressive the cancer will be, which results in a less chance of survival, so a worse overall survival rate. And it's relatively rare, but it's prominent enough to cause 400,000 deaths yearly internationally. And so specifically just looking at the US, in the samples that we looked at with this particular source, 23% of the samples had the mutation of the P53 gene. And so as I mentioned before, it's relatively rare, but in terms of cultural differences, the mutation is more commonly seen in African American women as opposed to white women. So some of the risks include developing different forms of cancer. So for example, ovarian cancer can come from having breast cancer. And with the P53 mutation, there's just a higher chance that the symptoms will be more severe, causing the higher death rate. So like I said, with the cultural differences, we're now looking at age differences in the cultural identities. And so with white women, they are more likely to develop breast cancer over the age of 55. And then as opposite, African American women are more likely to develop breast cancer under 45. So not only are we seeing two different cultures, we're seeing two different ages and how and where they develop breast cancer. And so the prognosis with breast cancer is, with the P53 mutation, is associated with TNM, which is looking at the tumors, the nodes, and the metastasis, and then the rate of disease-free survival. So with the mutation, the disease-free survival rate goes down significantly because of the expression of the mutation. And like I said before, it's just low survival rates with the mutation because of how severe it is. And so the patient can be expected to have very severe symptoms, as well as a decrease in overall health. And I mean, pretty much it ultimately results in death.
So there are some controllable and non-controllable factors that come with breast cancer. So some of the factors that one can control is weight and diet, exercise, smoking and drinking, and use of birth control. And some of the factors that you can't control that might contribute to your probability of getting this kind of cancer is age, gender, family, and your personal history, genetic information, and your race. So there are a couple different treatment types for this kind of cancer. One is surgery to remove the cancerous tissue. Another is chemotherapy, radiation therapy, and there are also clinical trials. So specifically for the P53 mutation, there's PRIMA1, which modifies core domain of mutated forms of P53, APR246, which induces programmed cell death by reactivating P53, PK11007, which is an anti-cancer activity focused on the P53 compromised cells, and COTI2, which is a tumor suppressor gene in clinical stage orally available.
These were just like other forms of chemotherapy that we found that were more specific to the mutation as opposed to just attacking breast cancer in general.
So with chemotherapy, it targets cancer cells and their ability to reproduce in an uncontrolled way. So this drug aims to impair mitosis and stop the cell division in order to control how much the cancer is able to grow. So chemo may also target the enzymes or hormones that the cells use to grow. It targets active cancer cells, and if the drug damages healthy cells, then side effects can occur. Radiation is typically the chemotherapy for breast cancer, and normal cells are still susceptible to the effects of radiation, which is why sometimes when you're going through chemotherapy, you can see a general decline in your health because your cells aren't able to reproduce to keep you functioning normally like they usually are.
So with chemotherapy, there's a bunch of risk factors that come along with it just because it's such an intense treatment. You can get things like fatigue, vomiting, mood changes, organ problems, trouble breathing, almost anything you could think of, infections, appetite changes.
And the organ problems is where different forms of cancer can show up.
So with chemotherapy, the side effects and risk factors can fade pretty quickly after treatment ends, or they can last longer. Side effects can either be short-term or long-term. And with normal cells, they can be damaged by chemotherapy, but that's just the risk they take to get rid of the cancer. And a lot of blood-forming cells in blood marrow, hair follicles, cells in the mouth, digestive tract, reproductive system, and a bunch of other organs will get affected by the chemotherapy. So with genes and oncogenes, TP53 gene provides instructions for making a protein called P53, which is a tumor suppressor gene, which is the mutation that we're looking at, and that regulates cell division by keeping cells from growing and dividing uncontrollably. And oncogenes are produced when there's a disruption in the amino acid pairing, and the P53 mutation is the result of this disruption, which leads to the cancerous cells. And with the cell cycle, the P53 gene triggers apoptosis in the cell cycle control. The mutation continues cell cycle when it should be repairing cells and going through apoptosis, which is in the gene 1. Cells can become cancerous because of this.
So the mutation causes the cell cycle to continue as opposed to stopping and repairing the cells, which is why cancerous cells can form from like damaged cells that aren't being repaired during the G1 phase.
So when we look at the causes of cancer in the early stages, especially like the G1 and the S stages, we see that cell cycle regulators are the cause of this. And then if we specifically look at the G1 phase, this is where the P53 mutation is located. And this occurs when the regulator does not become recognized or does not recognize a mutated gene in the G1 phase. Therefore, it's able to get past the G1 checkpoint and continue the growth. Yeah, the growth, and that's what overall forms the tumor that we see in the breast. And the mutation continues to replicate into the S stage, and this is the result in the cancerous cells that we see. So when we look at the inhibitors and cyclins, the cyclins drive the events of the cell cycle by pairing with CDK enzymes, and they signal that it's ready to continue to the next stage. So when we're looking at ours and the P53, it's able to get past the CDK enzymes of the G1 checkpoint, and that's how it's able to continue its growth. So the mutations in the P53 gene trigger the production of the enzymes, and this doesn't allow the cell time to pause in the G1 phase, and that's normally when it repairs the cell. So since there's no pause time to repair, it just continues, like we said, and it metastasizes. And this affects the regulatory loop of the regulatory cells, and it causes the damaged cells to build up and then become cancerous and form a tumor.
And that's how you form like solid tumors with the P53.
So when we're looking at mitosis and the P53 gene, mutations occur when a regulatory stage of mitosis is performed incorrectly. So when we see that they start pairing up, there will be one area where it's not correctly paired, and it's able to get past that G1 checkpoint and continue replicating. That's when it's able to get past the mitosis stage and it's not performed incorrectly. The mutated cell is able to rapidly produce other mutated or cancerous cells. And this is when we see 50% of human cancers is caused by this misproduction of the P53 gene. So here we have our Punnett square. The mother was heterozygous and the father was homozygous. So we have the Punnett square. And so the mutation can prevent the function of the normal protein from the unmutated allele.
Yeah, so there's about a 50% chance of getting like homo and hetero. And then here's our information, and then here's our pictures. We're done.